That's it. Hey, good evening, everyone. My name is Andy Page. I'm President and CEO of Oak Ridge Associated Universities. And I want to welcome you guys here to your graduation ceremony after some hard couple of weeks here in Oak Ridge and doing some great things. And we get to recognize those achievements tonight. I particularly want to recognize Jennifer Harper Taylor, who's here as the president of the Siemens Foundation. Jennifer, you always seem to be able to come back every single year, this time of this year, and spend some time with us. And it's a great pleasure to have you back with us here again tonight. I just wanted to recognize you as part of the foundation that helped sponsor and make all this possible. But we're here tonight to talk about you and your achievements as outstanding educators and researchers. And you had the opportunity here to take back to your classrooms what may change the future for your students. And you had the chance to experience some of the rare experiences of working in a national laboratory, probably one of the most premier science laboratories in the United States. I think a rare experience for all of us who gets a chance to go out and spend time at Cornell, but specifically with you who get a chance to improve your curriculums and take back into your classroom what you've been able to learn here. So it's a pleasure to be able to recognize your accomplishments tonight, and I want to thank Oak Ridge National Laboratory, Dr. Tom Mason's with us here tonight, who also speak to us, but they, in cooperation with us and the Siemens Foundation, make this possible every single year. I would like to say a few words about Dr. Mason. As the laboratory director, he's responsible for a diverse portfolio of science and energy, research and development through physics, biology, chemistry and climate change, energy generation, distribution, end use, and national security programs. I think we can safely say that Dr. Mason is at the head of solving some of the most complex scientific problems we have in the United States today. He has a Bachelor's of Science in Physics and a PhD in Physics from McMaster University in Hamilton, Ontario. Please help me welcome Dr. Tom Mason up here for this evening.
Well, congratulations to all the STARS Fellows. Thank you, Andy, Oak Ridge Associated University, Siemens Foundation, the College Board, and Discovery Education. And I'd also like to thank the staff from the lab who've been mentoring and providing support. So this is the 5th summer for STARS, so I think we can say this is definitely a strong program, and we've got some really motivated teachers over the years. I hope your time in Oak Ridge has given you a sense of the scale and scope of our research portfolio, and also maybe a sense of how we do our work. No, it's not a brilliant individual sitting in splendid isolation. It's a team sport, and that's part of what makes it fun. And hopefully, if you can communicate a little bit of that back into your students, it'll give them something to think about as they try and understand what their career options are and how science and engineering works. Certainly educating the next generation of scientists and engineers is an important issue for ORAU and the nation. Discovery and innovation are really the foundation for transforming our systems for producing and using energy, hopefully in cleaner, more cost-effective ways, enhancing our security and making sure we remain competitive in a very tough global environment. Now, in the 20th century, the U.S. led the world in putting innovation to work, and it's no coincidence that it also led the world in terms of the level to which it was educating its citizenry. We're no longer quite as unique in terms of raising our population to higher levels of education, and that's one of the reasons that we're in a more competitive situation in terms of the economic and industrial drive to support our standard of living. Other nations are catching up. They've increased their investment in both science and technology and also STEM education. And we should be pleased it's raising the standards of living of millions, even billions of people around the world. But at the same time, we need to respond. We need to keep a well-educated workforce to compete. And STARS is one way to help do that. Middle school and high school are obviously important components of the pipeline. And of course, it's not just about scientists and engineers, but it's about a population that has an understanding of science and engineering, since so many of the challenges we face rely on at least some level of understanding, whether it's, controversial topics like climate change or some of the health research related topics, and without having some understanding of the science behind it, it's easy for people to get swayed by, what may be kind of ill-informed rhetoric. It occurred to me at dinner talking about trying to get all the data at 250, that maybe Discovery Education should view this program as an opportunity for a new reality program. It sounded a little bit like, what's the Bravo show with the Project Runway, right? The deadline is coming, you've got to get the clothes all pasted together. I don't know if it quite had the audience, but I guess that's something that people who know that business would have to decide. But even if it's not a good candidate for Project Runway, I think this activity is something that can hopefully take a little bit of inspiration back to the classroom, because getting students excited is really critical to develop talent and need. And as I said, it's not just about scientists and engineers, it's about entrepreneurs, it's about communicators and just generally informed population. So thank you for your commitment to STEM education, for choosing to spend part of your summer with us. I hope you've enjoyed your time at ORNL and also in East Tennessee, and I hope you've built up some relationships that will last longer than just the short period of time that you were here. You're alumni now, you know one another, and you also know your mentors here at the lab, so I'd encourage you to stay in touch. That'll really help give you the full value of the experience. So best wishes as you return to your schools and take the word back. Thank you.
Well, good evening, everyone. Let me introduce myself. My name is Dean Avatius, and I am the Director of Science Education Programs here at Oak Ridge University. I want to welcome you all here to ORAU tonight, and I hope you've had an enjoyable couple of weeks. I'd like to extend the welcoming remarks from Andy and Tom, and to acknowledge some of the other distinguished visitors we have with us. In addition to Jennifer from the Siemens Foundation, we also have from our friends at Discovery Education. I'm looking for them out there. Kate McNally and Rob Warren. Where is Rob? Where are you, Rob? I saw you earlier. Has he disappeared on us? Okay. Well, we're very pleased to have you both here with us tonight. From Oak Ridge National Laboratory, in addition to Dr. Mason, we also have a number of the mentors and scientists who have made the scientific work possible this summer. And it really is no exaggeration to say that without you, this program could not work. So I'd like, if you would please indulge me in standing up so that we can acknowledge you with a round of applause for all your work this summer. So please, mentors and scientists, can we get you to stand up? My understanding is that maybe we have a few family members here from the educators. Can we give a hand? Do we have any family members here? Yeah, here to support your family members, and we're pleased that you can be here tonight. Okay, we're going to be moving into the main part of the program shortly, which is to hear from you. So I'm going to introduce you to Dr. Julia Abbott, who is the project manager here at ORAU for this program. Before coming to us at ORAU, Julia earned her PhD in chemistry from the University of Tennessee, so she's a fellow STEM person. What you may not know about Julia, in addition to being a chemist, she spends a good deal of time curling in her time away from work. And I don't mean curling, I mean out on the ice curling. In fact, there is a curling community here in East Tennessee, which I had no idea existed until Julia brought it to my attention. And there may be, how many are there, Julia? 5 of you in all of East Tennessee? But yeah, so if you want to know more about curling in East Tennessee, talk to Julia about that. Please join me in welcoming Julia.
Right, yeah, too bad we talked to Dr. Mason about curling. You know, he's Canadian, so there's a lot of us.
Okay.
I just had a couple other people I wanted to thank tonight. First of all, the facilitators who live in the hotel with everyone for 2 weeks and drive the vans and deal with all the problems. Not that there were any problems, but if there were, they would have dealt with them. So if Jim Davis and Jackie Collier and Kelly Rainey would stand up, and I would like to do. If I'm not mistaken, you guys have been with Stars all 5 years, right? Around a long time. And also I want to thank, well, she's not in here, but Paimua, who is my right-hand lady, and she's out there doing something. I don't know. But she's wonderful too, and we're going to applaud her. All right, so I just want to thank you all for a great STAR session. As many of you know, this is my last program at ORAU, so I'm very sad to leave, but I'm glad that I had a great group for my last ones. So now that we are going to start our group presentations, I'd like to welcome the first group, who is in the Chemical Sciences Division, the Special Projects Group. Their mentor is Barbara Evans, who's here with us tonight, and Kelly Ramey was the facilitator for that group. So you guys will join me on stage?
Well, I'm Corey Howard, and these are three of my colleagues who have to work together all week long. And I think Dr. Abbott was unaware that I was going to speak first for our group, and I think had she known that, she would have thought long and hard about putting our group up here first. So anyways, we worked with Dr. Barbara Evans all week, or the last 2 weeks, and we worked on cellulosic biomass reduction for molecular visualization by neutron scattering. Biomass creates a great potential to come up with biofuel and bioproducts, but one of the things that we're running into is the cost and the efficiency of that process and what it's going to take to bypass or get over this major hurdle and what we're trying to accomplish with that. Essentially how it works is biofuel comes from the product, the plant mass, which we want has to be converted to sugars, and then from there it's fermented into a bioethanol. And so where we run into an issue is early in the process where we can't get to the sugar fermentation because of some of the barriers that we encounter. So we're trying to get over this hurdle right here in an efficient, timely manner that can be executed. So we have the plant mass, and essentially what we're trying to do is take the plant mass, break it down, and then get it fermented. Like we said before, we're stuck in this process, and the reason that is we have this hemicellulose that we need to remove. It acts as a laminate as far as trying to get to the cellulose. So in order to make that happen, we got to take that away. But what happens is the lignins come in and they act as a barrier, kind of like a fly trap type process where they start to bundle up. In a sense here, and the cellulase can't get to the cellulose, and there we're stuck in the breaking down into sugar process. And so we spent a lot of time on trying to figure out how we can better understand the lignin and how it works with the cellulose, and how we can better get over that hurdle that is causing issues with this the efficiency of turning biomass into biofuel. So one of the things that we wanted to do is to look on a molecular level how lignin and cellulose interact with each other. And in order to do that, we use some small angle neutron scattering. And and so what needs to happen is we need to use deuteration, and by using D2O, you can see that we start to, the more D2O that we use, we get a better visual of what's going on. The problem is as great of picture and understanding that we can get in this process, the plant doesn't like to work with the D2O. It's a problem getting the plant to take it in. It's a problem keeping the plant without killing the plant. And so we're where we get from here, get a better visual, but also still maintain the integrity of the plant. So our goal this week was to get deuterium into the plant, and we tried to make this happen with the D2O, and we took up, we did a couple of different things. First of all, we introduced deuterium into duckweed, and the second thing we also looked at was switchgrass and the effectiveness of pre-treating switchgrass and also using that cellulosic enzyme for digestion, and also we have multiple variations where we use treated and untreated, trying to figure out that process. And the last thing we also looked at too is how is the plant reacting to the CO2 uptake and the photosynthesis activity of the switchgrass. And I encourage that we show a photo to better demonstrate how a plant takes up CO2 for understanding purposes, but the three ladies behind her vetoed that idea. So that being said, those are the things that we tried to tackle. Those are the things that we went after, and I'll turn it over, and they're going to talk about each one of these attacks that we encountered throughout the 2 weeks.
Hi, I'm Dawn Ramirez, and I'm from Fort Stockton, Texas, and I'm honored to be part of this. One of the first things that we did was look at some plant samples, and as Corey mentioned, duckweed or Lemna minor is the plant species that Barbara Evans is trying to use to try to get the deuterium into the plant materials to target the lignin. And you can see the reasons that duckweed is chosen. It's very fast growing. It has a very simple, or if you would, primitive root system, because one of the barriers is that you see that the plants will take the deuterium up into the roots, but it's not making it up into higher parts of the plant, the parts that would actually be used to produce fuel. They've been shown, or duckweed has been shown to take in amino acids, which is an important property. So. And another good reason for using it is, as you can see in the larger image, that it's very consistent in size of leaves. So it makes it very easy to do calculations on how much plant matter you're actually dealing with. And it's able to tolerate some levels of deuterium, and that's what our experiments, 2 of our experiments had to do with. All right, so in the first procedure, what we were doing is taking samples of duckweed and putting them in environments with higher and higher concentrations of deuterated phenylalanine. And what we expected was at the lowest level that the plants would survive and eventually begin to grow, and that we would see some inhibition at the higher levels. But as you can see in the graph on the left, our control plants both did very well and not only survived but thrived. But we saw some lag time where the plants were not really growing, just kind of hanging out. And then we started to see some growth. And then we took a 3-day weekend. The plants are well taken care of because, of course, Barbara was there the whole time. But when we came back to the lab on Tuesday, we saw that all of the samples in any level of the deuterated phenylalanine had died off completely. So Faye and I refer to those as the death jars. And they are not zombie plants. They did not come back. So so then we went on to Plan B. So in the second procedure, you can see that we had 6 different samples. And the idea there was to give them a little booster shot, put some glucose into the environment, and that would help to give the plants a carbon base so that they wouldn't have to rely so heavily on photosynthesis, that more of their energies could go into survival and even into growth. And we just set that up on Tuesday, so when you see we only have 3 days of observations. And again, surprisingly, the control plant with absolutely nothing but the water and the Hildebrandt and Shanks did very well. The one with the glucose did quite well. And right now we're kind of seeing that lag time again. They're kind of hanging out in the other concentrations. But again, it's only 3 days of observation. So this is an experiment that's ongoing. And the hope is that Barbara will have some plants that will take some lignin up so that she can see that and then she can pass it on and to the folks over in the second part of the project, which will use the plants with the deuterium in them for visualization.
Hi, I'm Kelly Weston, and I'm from Pittsburgh, Pennsylvania, and I was very honored to be here this week as well. So I'm going to talk to you a little bit about the digestion of the switchgrass and the procedure that we followed for that. First, we measured out 4 different vials of each sample that we used. So we measured out switchgrass which was grown in water, switchgrass that was grown in 50% D2O, and as our control we used filter paper because it's a great source of cellulose. We placed those in the vials, and we placed half of them in these little vials here. Half of them got pre-treatment of 1% sodium hydroxide, and we let those sit on the bench overnight. The other half we placed in the freezer to preserve them for the actual cellulase digestion. After 24 hours, we rinsed off the pre-treatment, added a buffer, and then placed the cellulase enzyme. We had matched samples, so half of the matched samples received the cellulase enzyme, and the other half did not receive the cellulase enzyme. Then we took 100 microliter aliquots of our digestion. We put those in the incubator at 40 degrees Celsius. And at 24, 48, and then when we came back on Tuesday, which was about 138 hours, we removed aliquots of those for analysis. What we did in terms of analysis is to find out how much cellulose was converted to glucose. We used an agent called a glucose assay reagent, which contained all of the materials that were needed to convert the glucose in the sample into a material that would absorb light at 340 nanometers. So we created glucose standards. We added the glucose assay reagent to all of them. If needed, when we took the samples, some of the samples, because of digestion, you'll see that when Faye shows you the vials, we had to centrifuge some of them to get out the solid material before we added the glucose assay reagent. And then we analyzed them spectrophotometrically at 340 nanometers. Based on the standard curve, we were able then to figure out the amount of glucose that were in each of the samples. So this is just a slide, again, showing the cellulose. And when we are digesting it, our goal is to get the glucose by itself using the cellulase enzymes. These were our results. These results are presented in a percent yield format. What we did is we calculated the theoretical mass of glucose that should have existed based on the evidence that 12% of the switchgrass is dry biomass, and that there's about 35% glucose within the switchgrass. We used some research for that. And we compared that to the mass of glucose that was experimentally determined to get a percent yield. The interesting part to note is this is the 24-hour digestion and the 138-hour digestion for water. You'll notice that the switchgrass grown in water, you really didn't get much of a gain of digestion, which pretty much means that in an economic perspective, 24 hours was plenty of time for pre-treatment. The other thing that's really interesting to note is that the amount of glucose conversion, or the cellulose conversion to glucose, is very similar in the water and the 50% D2O switchgrass. Again, very significant because if you want to use the switchgrass grown in 50% D2O to do scattering experiments and determine the molecular structure, you don't want to have any differences in how much glucose can be produced. We also have on here the untreated switchgrass samples, and you'll notice that even after 138 hours, we still haven't digested and produced as much glucose as we did in 24 hours with pre-treatment.
Okay, so With this experiment that we did, one of the things, these are the actual vials that we used for the experiment. And all the samples on this side did not have enzyme, the cellulose enzyme, which breaks down the cellulose wall, and all of these samples did. The white ones are the filter paper, which we used as our control. We had our switchgrass samples that Barbara had provided us that were grown in 50% D2O, and switchgrass that was grown in regular water. So the next thing we wanted to see is what happened to the plants visually. We were able to experiment with the plants and show that the cellulose was digested and turned into glucose, but we also wanted to see what the cells looked like. So we did some microscopy, and this first set, these are the switchgrass samples that were grown in water after 138 hours. So if you look, we had 4 different samples, and the first one, if you look at this vial, this one did not get any pre-treatment with sodium hydroxide, nor did it get the cellulose enzyme. You notice that the cells look just like normal plant cells. The sample that did get the pre-treatment but no enzyme, what we noticed with the microscopy is that the cells kind of got a little bloated. So you can see that sodium hydroxide probably had an effect on the plants itself. The next sample, we had no pre-treatment, meaning we didn't use the sodium hydroxide, but we only added the enzyme. And what you can see is the enzyme definitely chewed up some of those thinner cellulose cell walls. So you can definitely see that digestion. When we added both the pre-treatment and the enzyme, which is this last vial here, and you can kind of see it in just visually looking at it, but when we look at it on the cellular level, you'll see that the combination of the pre-treatment and the digestion gives you like really good cellulose breakdown. So this gives us evidence that the combination of the pre-treatment and the enzyme is very effective with plants grown in water. So we compared that with plants that are grown in deuterium D2O. So these are our samples that were grown in D2O, and we noticed similar things. This sample had no pretreatment, no enzyme. The cells look about what plant cells look like. We added this one with the pretreatment. You'll notice that there is some of the same kind of bloating of the cells. Here, this is a good visual of seeing what the cellulase enzyme did to the plants. You can actually see the cell walls kind of getting looser and starting to break down in the deuterated samples. And then again, in our last set of samples, you'll see that it's a full digestion with the combination. So this was it. With using the microscopy, we were able to get evidence of the usefulness of having both the pretreatment and the cellulase enzyme combination. So that's a basic overview of our project. We wanted to take a quick second just to talk about the things that we got from our project. And it was funny, just as was said in the beginning, coming in here, you think that science is all about just doing an experiment in a lab and coming up with ideas. But what we did learn is that it's about collaboration. It's about scientists talking to other scientists, teaching people things along the way, meeting people, these are my colleagues, that you can share ideas with and get really energized and good ideas. The lectures and the sharing of information that we had were really great. And with this experience, we also got a chance to get some history and view some nature, so we could kind of get a perspective on our world. So this was, for me, it was a really great experience. It gave me a lot to give back to my classroom.
This experience has been life-altering, I guess is a good way of saying it. It's a dream come true. When I went into education, and I went into it straight from high school, straight into college, I always knew that I wanted to teach science. I never really thought about the importance of doing research. When all my other fellow chemistry majors were doing internships at Bayer and PPG. I went and worked at the Carnegie Science Center as a science intern. I was on stage and I did presentations and I supervised the team volunteer carts and all of that wonderful stuff. And I never gave it a second thought as to how much I was missing in that research. And as I began to go through my career and get to the point of teaching AP chemistry, I really realized that I had an education gap that I really needed to fill because kids were asking me questions and I couldn't answer them. I didn't know what it was like to be a researcher. So when I saw this opportunity, I jumped at it. And to be selected was such a great honor. Besides the research, and like I said, that was first and foremost my main reason for applying, was the fact that I got to see a research team have a meeting. I got to see the collaboration and the respect and the ability to question without antagonizing each other and to challenge each other to do bigger and better projects. And it really opened my eyes as to the process of science. And I can take that back into my classroom and apply that knowledge. I met 19 other people that have, opened my eyes to all of the opportunities that are out there. It's nice to be with like-minded individuals. I'll call you my friends now, Definitely a Facebook group that we have, you're going to be hearing from me. And the great lectures. I mean, you get to hear a Nobel laureate talk and all the experiences and the tours and the field trips and the camaraderie was wonderful. But yesterday I had this unique advantage. I found out that one of my former students was here as an undergraduate intern. He had contacted me via email. His mom said, hey, did you know Mrs. Weston's there? And so he sent me an email. I got to sit down with him at lunch yesterday, and I got to talk to him about his experience here as an undergraduate intern and my experience as a teacher. And I realized as much fun as I'm having, it's not about me. It's about taking it back and applying what I've learned to the kids to inspire them to move on and to get to that level. And it really took a few minutes for me to realize, wow, it's not my life. This opportunity didn't just affect my life. It's going to affect every student that I teach from here on in. I think that was the greatest thing ever.
Well, for me, any good professional development is what I call fuel for my fire. Teachers can get burnt out during the course of the year with all the politics and things that we have to deal with. So aside from sharing, resources and knowledge with my colleagues and sharing experiences like the field trips and the tours, it's a booster shot for me. It inoculates me against complacency and routine. But this experience has been by far beyond anything I've done before. The opportunity to do some real hands-on research, which again, like Kelly said, I've never had the opportunity to do, with state-of-the-art tools, with an expert in her field, was an experience that I couldn't pay for. So that was free. I also saw the interdisciplinary nature of science. It's not something that's isolated. It is a meeting of the minds. It's interdisciplinary, it's international, it's between genders. I'm happy to see a lot of women in the labs. And when it comes to me being in the lab, I was in unfamiliar content, and it put me in the shoes of my students. As a learner in a content area where you're not familiar, you're already uneasy. But when you're talking about lignocellulosic biomass, recalcitrance to digestion in the presence of cellulase enzymes, I'm a second language learner. And if it wasn't for our two translators, Kelly and Faye, and Barbara's patience and willingness to explain and re-explain and re-explain, I would have gotten frustrated and felt in over my head very quickly and I would have wanted to give up. So I really, when I go back home, I've got to constantly remind myself where my students are. And the 19 people that I've shared this experience with, the researcher that I got to work with, the presenters, they're all inspiration to me, that when I go home, I'm an ambassador for STEM. And I'm going to take that to heart, and I'm going to take it to the streets. So that's my experience.
So my name is Corey Howard, and I'm a middle school teacher in Indiana. And research with 12 and 13 and 14-year-old kids is a little bit different than the research we do in the lab. The research assignments that I'm most familiar with is a controlled experiment where the manipulated variable is antiperspirant. And so when I got here 2 weeks ago, there was a lot of scientific content learning that I had to take on. And so it's a personal growth and a professional growth. And I'm in the same boat as Dawn was. It was nice when I saw my list. I said, there's another middle school teacher. I'm not alone here. And Siemens got it right when they brought Faye and Kelly in as far as their abilities. They really are talented teachers, and I can say that because they taught me as a young adult a lot about what's going on this week, and so I do appreciate that. And because of their efforts, I think Dawn and I are both a better teacher today than we were 2 weeks ago. We went to the Smoky Mountains, and Dawn and I, we went on a hike, and we were in the middle of nowhere for probably 1, 2, 3, almost 4 hours. And during the time that Dawn and I started talking, it just so happens that we both actually put on haunted houses at our schools, in a sense. And so one of the byproducts of this biomass to biofuel studies is the advancement of junior high haunted houses all over the nation. And so, Dr. Evans, you have my permission to add that to your annual report next year. The other thing that was really cool is we got to work with Kelly, and she was unaware that she was in the photo presentation, but that is a pen in her mouth, not a cigar. But it's so cool because she was a high school teacher, but now she's a professor, and she's as passionate about the students that she teaches, but the kids that are still in the education system, and as she is about her content. And so it's been one of the great resources that come about is I now have a professor that I can reach out to and ask for help or ask for insight, and have a professional relationship that's going to last for some time to go. And another great experience that I had here in Tennessee was I know that some people are making efforts to lose their Southern accent. And so, hanging out with Kelly this week, it was actually the other way. I had a 2-week crash course in obtaining that Southern accent. And so, I get to go home tomorrow with a hankering. And I'm not really sure what it's for, but it's definitely a hankering, so thank you for that insight. We met Dr. Barbara Evans last Monday, and conversations with her are definitely very insightful. And when you ask her a question, it normally starts off with a basically, and then she starts to articulate what's going on. And then about halfway through it, she's kind of, forget it. Let me tell you how it actually works. and I've done more in 4 minutes of talking to her than I did in 4 years of college. And I wasn't always sure what I was hearing, and so it's like the penguins of Madagascar. I just smiled and waved. And so that was just an amazing time to just learn. And I remember on day 1 feeling overwhelmed, in over my head, and clueless. And I thought to myself, I haven't been this clueless on day 1 since I was on my honeymoon. So I go back to Indiana tomorrow, and as a middle school teacher, I remind the kids all the time, hey, we're not doing rocket science. My eyes have been opened. That's the wrong quote. Okay, it's not that it's rocket science. It's, kids, we are not doing cellulosic biomass production for molecular visualization and neutron scattering science. And so, the time invested as me as a teacher from all the people involved, especially Dr. Evans, it is definitely going to have an impact on the school year. It's going to make a difference, an immediate difference in the classroom, in my school, and in my small rural community. I think I'm destined or set to serve as a leader in a STEM initiative moving forward because of those opportunities. And one of the things I've learned throughout this process is that, one person's research is building on knowledge that goes on to another person, and knowledge is building on knowledge, and indirectly everybody is affecting and having influence on the people that may came before them or after them. And so, as I'm here these 2 weeks and just a teacher helping out on research as part of the fellowship. But when I get to go home, none of you people will be around, and so I get to tell the story that I'm a scientist that did 2 weeks of research. And the more I tell the story, the more I probably talk about how great of a scientist I truly am. And so one of the lessons I learned as a great scientist, that the impact that I have here is in my classroom, but the knowledge is going to continue to grow, and I'm genuinely so appreciative of the time that Barb invested in me, because I know it's going to make a difference in my classroom, but it's going to make the largest difference in my home and how I raise my children. And so the lessons that I learned are going to have an influence on the outcome of my children's futures and my lives. And so, Barb, I thank you genuinely from the bottom of my heart that you took the time to invest your experience, your knowledge with us, regardless of what backgrounds we came from. And for all of us, anybody that takes the time to have an influence on our own personal children will always have a special place in our hearts for what you gave to us. So thank you.
Corey, I think the sounds taught you how to talk. Remember what I said about the,
I thought it was Kelly said that.
All right, so next we have another group from Chemical Sciences. They're in the Materials Chemistry group. Their mentor is Karan Karan, and he's sitting right over here too. And their facilitator is Jim Davis. So guys, come on down.
Yeah, there is wireless mic.
Good evening and welcome to our presentation this evening. My name is Megan Hart. I'm from Maryland.
Patricia McHugh, New York.
Jay Kreisman from Jersey.
Our group is the third teacher research team to work on and finish preparing the preparation of lithium titanium oxide, otherwise known as LTO, and a material from sustainably sourced lithium carbonate for the use in lithium-ion batteries. As an overview in our presentation, we will discuss the purpose of our project, some background information, the materials and method, our results, and lastly, future work to be done. The purpose of our project was to compare the effectiveness of lithium-ion batteries prepared from lithium titanium oxide from two different sources of lithium carbonate. We used a commercial source provided by Alpha Azar and an experimental source provided by Symbol Materials. A little background on lithium-ion batteries. Current lithium-ion batteries are used in everything from mobile devices to laptop computers, as well as renewable energy sources and electric vehicles. Some advantages as compared to older battery technologies such as nickel-cadmium and acid-lead batteries are that they are more compact, they have a higher energy density, as well as they have a longer capacity. Although they have many advantages, one of the important limitations is in reference to their safety and that they can cause fires. As can be seen in this diagram of a lithium-ion battery, the anode is made of carbon coated onto copper foil. The cathode is made of lithium cobalt oxide coated on aluminum, and between there's lithium hexafluorophosphate, which is an electrolyte dissolved in organic solvents.
So in this project, we replaced the carbon in the anode of the lithium-ion battery with an LTO. The advantages of these batteries over traditional lithium-ion batteries are that they charge faster and they are safer. There are some limitations, including lower voltage and lower energy. Uses for these LTO batteries will be military applications, electric vehicles, drones, and also situations in which safety is a priority. The Department of Energy has put together a list of critical materials. These are in high demand and low supply. The elements in red are critical. Lithium in yellow is near critical. Part of our project is to ensure that lithium does not join the critical list of elements. So currently we only produce in the U.S. 1,500 tons of lithium per year. There's only one active lithium mine in the U.S., and that's in Nevada. So we currently have to import 80% of our lithium. Some advantages of U.S. brine extracted lithium are our decreased reliance on other countries, and also we can sell the excess lithium to other countries as well.
Press the arrow.
Okay, this graphic shows where we stand in terms. This graphic shows where we stand in terms of lithium production. Although we are in the top 10, we fall far behind other countries in terms of lithium production. That's why we need to import most of what we need.
Okay. The start of the process is to get lithium carbonate to react with titanium dioxide. That gives you the LTO as well as carbon dioxide. We use two different sources for lithium carbonate. The first was a commercial source by the Alpha ASAR company. The lithium carbonate came from Argentina. We were able to verify that. We were unable to verify whether it was mined from an ore or extracted using a brine process. The second sample of lithium carbonate that we used was an experimental sample from the Siemens Corporation. That came from California using a brine extraction process. And this is the brine extraction process. It's basically a way to get geothermal energy when some of the brine is shunted into a simple operation where the lithium is separated and then the brine is returned and recycled. If this is successful, Siemens plans to partner with Tesla to help Tesla make batteries for its cars.
In terms of materials and method, the first part was to prepare the LTO. In order to prepare 5 grams of LTO, we followed a series of steps. First, we mixed lithium carbonate as well as titanium dioxide. We combined them using a mortar and pestle, and we heated for 30 minutes. We then heated our sample to 650 degrees for 6 hours. The purpose of this was to drive off any CO2 from our reaction. We then mixed our sample again and heated it, but this time to 800 degrees Celsius for a longer period of time. The purpose of this was to create the LTO, which was our desired product. We used X-ray diffraction to verify we synthesized LTO. The data from X-ray diffraction shows crystalline LTO, represented by the blue lines, with a trace amount of unreacted titanium dioxide represented by both the green lines, below here, as well as denoted by the asterisks. You can see that the commercial sample contains LTO, as well as the symbol sample. This XRD graph shows both the commercial and symbol samples superimposed to show that both sources of lithium carbonate produce significant amounts of LTO.
The next step in our process was to prepare a slurry. We mixed our LTO sample and mixed it for 10 minutes. We then added some conducting carbon and mixed for another 20 minutes. This material makes the batteries better conductors. We then added to a vial 3 things: PVDF, which is a binder, some of the LTO carbon sample, and some NNP, which lowers the viscosity. We then added a magnetic stirrer and let it stir overnight.
The final step in battery preparation was to apply the slurry to copper foil using a gauging tool on the right there that puts a precise but even coat along the copper foil. Then we baked it overnight. And Reem Chow, who's in the audience, helped us with this. He's working in an argon glove box, and that's where he actually assembled the battery. Okay, in the glove box, he took the component parts, which you see on the right and on the left actually, and combined the can with the copper foil, coated with the slurry, using a porous separator, some lithium, and a wave spring that actually makes sure it ensures contact. I have a battery with me. You can see it's very, very compact and ideally very, very energy dense. And that's really the whole goal. The final step is to subject the battery to repeated charging and discharging to determine its characteristics.
This graph shows the battery charging performance for a lithium-ion battery without any LTO. You can see that as the battery is charged and discharged, over time it starts to lose some capacity. In contrast, this graph represents a lithium-ion battery containing LTO. You can see that this battery does not lose capacity over the time of this study. Battery testing for our samples will not be complete until the middle of September.
Once that battery testing is complete, the technical report will be released later in the year. And then LTO batteries will be commercially produced for use in electrical vehicles and other applications.
So we were admonished to keep the presentation as short as possible. All right? So when it comes to the takeaways, I truly speak for all three of us. And I gotta tell you, Corey's gonna be a real tough act to follow this part. But we did experience the value of true collaboration. I think what was lost in this presentation was the fact that it actually took all three of us to get that, what would become the slurry, into a 20-millimeter scintillation tube. One person was holding it, one person said, no, you've got oil on the side, you've got to fix that, and we did. And we nailed it. We nailed it 4 times in a row, which we're all very proud of, actually. Secondly, we saw the benefit of working in a scientific community. Just listening to people talk in the lunchroom is unbelievable, truly. We did feel the satisfaction of contributing to a larger energy solution. And we do plan to continue our collaboration by bringing electrochemistry units into our classrooms and hopefully making simpler versions of the batteries with our kids as well. I would like to sincerely thank the Siemens Foundation STARS program, Discovery Education, ORAU, ORNL, the Critical Materials Institute. Give a very special thanks to Dr. Franz Verankemann, Mr. Jim Davis, Rin Chow, and Vincent Liu for their help and support. And the last thing, just one last thing, that for me personally, that I think is unique to Oak Ridge. When you work at Oak Ridge, you feel like you're part of an important past as well as an important future. And I think that's unique to the place.
That's more like it. And one thing I want to do before the next group comes up is say one other thing about Franz and Barbara. I'm pretty sure that they have been a mentor every year since this program has started. All right? All right, so 5 years of STARS teachers has come through your groups. So I think let's give them an extra hand. In the room, you have a lot to live up to, 5 years in a row. And this year they did 2 groups. Anyway, so our next group, sorry, is in the Biosciences Division, the Plant Systems Biology Group, and their mentor is Sean Yang. Yes. Somebody needs to walk around the stage a little, I think.
I'm probably loud enough that I don't. Can you hear me, Josh? Yes, we can. Oh, it's
Right, good evening.
There is a microphone.
Just so that both of them.
I got it.
Now Derek will perform soft shoe. Good evening, my name is Chris Brothers, and I'm from Massachusetts, and I'd like to introduce my fellow researchers, Derek Berger from Maryland, for say 25 times in the past week, Catherine Hoover from Texas, and Rhonda Smith from just down the road here in Tennessee. And our project was looking at the possibility of bioengineering CAM photosynthesis into populus, which is poplar tree or cottonwood tomato, to improve plant water use efficiency. So back in 7th grade, photosynthesis probably seemed rather simple. Plants take in carbon dioxide and water, they produce sugar using sunlight, energy, and they give off some oxygen. And then hit high school and things got a little bit more complicated. And then hit Oak Ridge and it got really complicated. So somebody before mentioned carbon dioxide. Corey, here's a picture for you. How plants take in carbon dioxide. And they do that by opening these little pores on the underside of the leaves, which are called stomates. In the picture on the right, you can see some that are open and some that are closed. And I'm very, very proud of the picture that's on the left because that's one that we actually took under the microscope in our lab group, which I think is just astonishing. So the plants are taking in carbon dioxide, but the problem is that if you open your stomates to take in carbon dioxide, you also are losing water through evapotranspiration. So there's kind of this constant catch-22 for plants, whether to take in CO2 and give off oxygen or try to conserve their water, and this becomes especially important if they're under some kind of water stress or if they live in a place that is kind of a hot, dry climate to begin with. So this creates some real problems for plants. So most plants do a type of photosynthesis called C3 photosynthesis, and it's called that because, oops, I don't have the pointer here. It's called that because the first molecule that's produced during this process has 3 carbons. But there are a couple of other versions of photosynthesis, including C4 and CAM, and we're going to be looking specifically at CAM. So on this slide, you can see some examples of some C3 plants. At the top is soybeans and below are rice. Both of these are C3 plants, very important crops in terms of food. And possibly not coincidentally, both of these plants have also been genetically engineered. So what's different about a CAM plant? In a CAM plant, the plants open their stomates at night, and they take in carbon dioxide at night, and they fix that carbon dioxide using an enzyme called peptase, which Derek's going to talk a little bit more about in a few minutes. And they store that carbon dioxide overnight in their vacuoles in a chemical called malate. And then the next day, they close their stomates. Now they can conserve water when it's hot and dry out and sunny. And they convert that malate back into carbon dioxide and take the carbon dioxide off. And they run it through the Calvin cycle the way a C3 or a C4 plant would do. The advantage of all this is that they can conserve water. And so typically you see C3 plants have somewhat higher photosynthetic rates, but they use a lot of water. CAM plants, their photosynthetic rates are lower, but they are very water conservative. They might use only 20% of the water that a C3 plant might use. So some examples of CAM plants: pineapple, cactus, agave, and our hero at the top, Kalanchoe. CAM stands for crassulacean acid metabolism, so that's quite a mouthful, but it comes from the name Crassulaceae, which is the name of the family that Kalanchoe and some other plants belong to. It turns out about 36 families of plants use CAM photosynthesis. We didn't know how many families of plants there were, so we looked it up, and there's about 600. So really that's not too many plants. About 6% total can use CAM. But those plants are a real advantage in hot, dry climates. You might get the idea from the plants that are here. So what this project proposes to do is take the genes that control CAM, that whole metabolic process, and put them into a C3 plant, which would allow us to kind of capture the best of both worlds and have a plant that can have fairly high productivity, but at the same time be very water conservative. And as well, that would allow potentially growing some of these plants on what right now might be marginal lands where C3 plants really couldn't grow too well. And also, long term with climate change, if we see hotter, drier climates, we may in fact need plants that are better adapted to being able to live in those types of climates. And I'll turn it over to Derek.
Okay, so what I would like to do is briefly review the process that our research group is basically performing. Know up front that as we go through the rest of this presentation, given the long-term, the amount of time it takes to complete a process like this, a lot of the things that we're going to be demonstrating for you tonight, or showing you tonight, and talking about tonight, basically are simulated versions of the research that's being done in the actual lab. Either way, so the process that we're going to follow, the team is going to follow essentially and try to achieve the goal, is to first of all identify the genes related to CAM photosynthesis. If you're going to do bioengineering, you need to know what those genes are, the proteins that they make, and also, very importantly, the functions of those proteins, or the operations of those proteins control. The second step will be to engineer a plasmid. And so as a physics teacher, and I had no idea about any of this, and so this is biology from a physics perspective. So a plasmid is basically a package of DNA that will obviously contain those CAM genes that are identified, and then the plasmid can be taken and placed into what's called agrobacteria. Agrobacteria, some of you may be familiar with because you might have seen these like trees that have these growths near the roots. They look like kind of like globs of bacteria. Those are agrobacteria, and so agrobacteria is just ideal because it's a very stable platform for delivering a package of DNA into a C3 plant. After that, obviously the transformation has to happen, and so we'll use the agrobacterium to insert CAM genes. Once the CAM genes have been inserted into the C3 plants, we'll culture them up, obviously to grow these new organisms. And then lastly, there will be some testing done to make sure that the genes are actually expressing themselves in those new plants. And then as a final step, as Chris was saying, you want to know whether or not the plants are optimizing their water usage. And so you'll compare basically the photosynthetic processes before the transformation to the photosynthetic processes after, the CO2 uptake, to determine whether or not the plant is in fact optimizing the water usage specifically. And so the first step in this larger process is to figure out what DNA you need and what proteins they create and how CAM actually functions so you can create this plasmid so it can be engineered into the powder. And so the actual process of determining the DNAs and the proteins and their functions is somewhat complicated, and also traditional methods are, for lack of a better word, time-consuming. And so modern technology in the form of computer algorithms and computer programs give the group and scientists the ability to model, take protein structures, which are very well, sequences, excuse me, which are very well known, we know a lot about them, and using computer algorithms, which are kind of shown here in the diagram, and I won't go through step by step how it works, but using a computer algorithm, you can basically go from the very well-known sequences of amino acids in the proteins and transform those into three-dimensional models of what those proteins then look like. The really nice thing about the models is then from the model you can predict the function of those proteins. And not only can you model the function, but also the actual dynamics to see what that protein actually does. And so this is just a couple of visuals of what the end result of this step actually look like. On the right, what you see are three-dimensional structures of Hep-C, which is that critical enzyme in the CAM process that actually fixes the carbon dioxide at night when the stomates are open. And so you've got two different views, obviously rotated. Really interesting for me is we were walking through the visitor center and there was a picture like this on the wall on the first day that I just thought was like modern art and really cool. And by the third day, we actually got into the lab and we started learning about all this. I went back for lunch and I was like, oh, now I know why that's on the wall. Anyway, and so, for the non-biology people, you have all these spirals, each representing a given chain and structure of protein, and then in Hep-C, what they do is they all overlap with one another. And so from a three-dimensional structure like this, what you can basically do is predict exactly what this structure is doing within the cell, and then identify the DNA that goes along with it to put into the plasmid and engineer the plasmid and the agrobacteria. One last thing, the other thing that's really nice about this is not only do you get the structural shape, there's also software that allows you to get the dynamics. And so they've actually modeled the movements and the actual activity of these proteins, so we know exactly how they operate, what they do.
Okay, let's see if I get her in. Okay, so I want to show you some of the things that we did during the course of the 2 weeks. This shows us actually transforming Agrobacterium with GFP and M-cherry plasmids. And what that means is, on the left there, that's a plasmid. A plasmid is basically a circular piece of genetic information. And it's such that we can replace part of it with either GFP or M-cherry, which are genetic markers. And you can also put whatever gene of interest you want into that plasmid. And so what we're doing there is taking the plasmids and putting, infiltrating them into the agrobacterium. And then we take that solution, that agrobacterium solution that's been transformed, and we transferred it into tobacco plants. So we put them on the back of the tobacco leaves, and then we waited a couple of days, and we were able to check and see if our markers showed up. And that's what we found out. We found, if you look in the top left picture there, that those two little green dots, those are the nuclei of a couple of cells showing the GFP marker. And if we see the marker, we know that the gene we're interested in also made it into the cell. Also, we thought these were kind of interesting. The red that shows up here, these are the chloroplasts from the cell, which have their own natural fluorescence. And on the bottom right, you can see a picture that contains both those two nuclei plus the chloroplast. Okay, another thing we did was we prepared media for tissue culture. And so that's what we're doing here. We're preparing media, and then we take that media and we grew We sterilized and cut Kalanchoe plants and tissue cultured them. So the first 3 pictures there are us working on getting our little tissue culture made, and we were very proud there, so we posed with our tissue culture. But the proudest moment was this morning when we saw that if you look on the right, our tissue culture had actually started to sprout. You see 2 little sprouts on there. And so that's how we grow the little plants. And these plants are sterile and can be used then for genetic engineering. Okay, so once you have plants, we took the leaves and we infected them with agrobacterium by wounding them. We created little cuts in them, and then we covered them with the agrobacterium. And then over a course of a couple of days, in the top there you see the agrobacterium growing around our leaves. And if we leave them, which will happen in the weeks to come, they'll actually start to grow a new little plant. And that's what you see on the bottom there. And hopefully someday they'll look like the ones on the right.
Okay, so after we get that callus and all of our newly engineered plants, we're going to want to test to make sure that the transformed plants or the engineered plants are actually doing what we would like them to do. So one of the ways that we can do that is extract the RNA. You see there that we're working with the plant material that's kept very cold. That's why we've got the liquid nitrogen, and it also makes us feel like mad scientists. And we put that in a lysine solution and spin it in the centrifuge, and we get out our, we extract out our RNA. Here we used agave. We would, in the CAM project, they would use whatever plant of interest they're looking for. And then we had to take it to the nanodraft to verify that we actually had indeed extracted the RNA. And Kathy was our winner with the concentration that we were looking for. So the rest of us maybe didn't use enough quite plant material, but she really globbed it in there, so she got a good concentration. Then we want to take that RNA or DNA and do, Then we did qPCR, okay? So we take the RNA and we want to see if that gene of interest is being expressed. So you see here we put it into this machine, which was called the thermal cycler. That's what it was. It cycled through heating and cooling cycles. And this is actually what helped us to grow or to create more and more RNA so we could use it. And we had 3 samples dealing with the circadian rhythm. So once we got our results on the next day, the computer did its magic overnight, and it made the math teacher and me say, "Yay, graphs!" So the computer put a threshold on these curves on the left. The curves on the left are showing how many of the genes how many copies are being made of the genes, and over the cycle times. And then the graph on the right is showing the concentration. So you see the green at the top and the blue below that. That's showing that we had different concentrations of the gene, and that would make sense because since it's dealing with circadian rhythm, it was taken different times of the day, so you would expect to see that gene expressed at different concentrations. Then we ran it through a gel, electrophoresis, and we found out that we did, in fact, extract the RNA that we were trying to extract. So another way that the CAM project may check to see if their plants are doing the things that they actually want them to do would be to use a CO2 gas analyzer. So you plant it on the leaf, and this measures the amount of CO2 being exchanged in the air right around the leaf. And we can tell how much photosynthesis is going. And we can also check on the stomates, whether they are open or closed. So in our experiment, we saw that the blue dots there are actually the C3 plant, the poplar, and the red dots are the clusia, which is a cam plant. So as expected, the poplar is photosynthesizing at a greater rate, and the stomates are open during the day, where most of the stomates for the cam plant are closed during the day. Now, for the CAM project, they, in their genetically engineered plant, they're hoping that they would test their new plant in good times, so when it has plenty of water, and they hope to see that it's acting like a C3 plant. And then they would also test it in drought situations, and they would hope to see that the plant switches into a CAM photosynthesis and conserving water. So our classroom impacts. For me, in the mathematics classroom, I got to see a little bit of the way that mathematics is actually used in a real-world setting in a science lab. So that's just more that I can communicate to my students how it can be used. Also, it was eye-opening to me, not being from a science background, the scientific process. And I think with STEM, the whole idea is getting kids to think innovatively and to persevere and solve problems. So I think seeing scientists do this helps me to communicate to my students how to do that as well. And finally, understanding the biology better, because it's been 25 years since I studied any biology. It's going to help me collaborate with my fellow faculty at Heritage High School.
Yeah, so for me as a physics teacher, the biology portion was also eye-opening because I was not aware of all the things going on in microbiology at all, having not taken biology since 10th grade. And so what I'm going to take away are a variety of different things that I can share with my students, including the multidisciplinary nature of all science research. Nobody is operating, as somebody said, in isolation, working on one topic with one conclusion, one time, and then forgetting it after that. And I think sometimes in a science classroom, middle school, high school, we do those sorts of things and we leave them behind, and it's not elaborate multidisciplinary types of research, and so it'll be nice to convey that to them. Also, as an engineering teacher, the bioengineering process was very new to me, but it's nice to see a different variety of the engineering design process being put to work. And so it's going to be nice to go back and have this project as evidence of how engineering is done in a lot of different fields. Of course, the interconnectedness of the theory, processes, and skills done in science is something that I teach, but now having seen it in first person, it's very easy to explain how that works. And then teamwork. STEM is all about, in a lot of ways, teamwork and collaborating with one another in interdisciplinary ways to make progress that we wouldn't be able to make on our own.
Okay, by far my greatest takeaway from this is going to be the time that I spent in the lab. I'm a science teacher, but I'm not a scientist, and I've never been a scientist. And so having this experience gives me that to bring to my students. What it's like to be in a real working lab, and how the scientists work together. I mean, how our mentors work together, and perhaps that the vision that I have of a working science lab, and I know the vision my students probably have, are very different. So to be able to share with them that is probably the biggest thing for me. Also, I would like to kind of change the way I do some of my labs in class to kind of model the way that real science works, a little more so than some of the cookbook labs that we sometimes do. I'd like to encourage a little more collaboration and thinking from my students. So, and then also an increased knowledge of STEM jobs, because my students are graduating seniors, and actually many of them are AP students, and so the likelihood of them going into a STEM field is there. And so I've learned a lot about what's available for students in STEM, and I'll be really happy to be able to go home and share that with them.
So I teach AP Biology, and I just listed here a few of the things that are in my curriculum that we learned about this week. And if I conservatively look at this, I would say this is probably 60% of my curriculum, which I teach over 10 months, that we learn about in 2 weeks. So I want to give a big congratulations to my fellow researchers because this is not easy material that they were doing, and they've all expressed to you that in biology they took a long time ago. So they really, really worked hard this week to learn this and to understand this and to put this presentation together. And I think from my perspective, this answers the question that students always ask, well, why do you need to know this? Because people are using this in the real world and this is going to impact your lives. How do we learn all this anyway? Well, now we know the kind of research that goes into answering these questions. And then finally, I do actually a fair amount of research with my students. We have a very strong science fair, and I can see a lot of places where students could take on little pieces of this project to do as research projects. I don't think they're going to be genetically engineering poplar trees anytime soon, so I don't think you have to worry that we're going to scoop you. But so we'd like to just take a moment to thank our research group. And even though we had one mentor, we really had about 9, I want to say. We worked with probably everybody in the lab. And also to thank Siemens and Discovery Education for sponsoring our 2 weeks here at Oak Ridge.
Okay, so our next group is in Material Sciences and Technology in the Mechanical Properties and Mechanics group. Their mentor was Edgar Lara Cruzio, who's in the back. Where? And Jackie Collier was their facilitator.
Hi, I'm Shannon Pizzina. I'm a chemistry teacher in Alexandria, Virginia.
And I'm Josh Holt. I teach math, and I'll be moving to Chattanooga this year for being in Knoxville for a long time. I'll be teaching at Ooltewah High School.
Good evening, I'm Jeff Steinert. I teach physics at Arizona School of the Arts in Phoenix. I'd also like to acknowledge the fact that Chris Stevens, who is instrumental in our work, is seated back there next to me. I'm going to begin by covering some basics about the structure and properties of metals and alloys. Metals typically have one of these 3 common crystal structures shown here, BCC, FCC, or hexagonal cubic close-packed crystal arrangements. And when we step back a little bit and look at the structure of a crystal, what we see is that those units are repeated over and over again throughout the larger crystal. This, however, is a perfect crystal, and most crystals don't look anything like this. Real metal crystals contain certain defects. Among those defects are point defects, like the 3 types that are shown here, a vacancy, where there's a missing atom, or a substitutional defect, or something other than the atoms that surround it is included, or an interstitial point defect, where there's an extra atom wedged in between the crystal. The defect, the line defect, though, is one that we're mostly interested in here, and this is a dislocation. You can see that there's kind of a partial line of, or sheet of extra atoms that's wedged between the others. Here, and it doesn't continue down along the line. When a tensile stress, this is we measure stress by doing force per unit area, the cross-sectional area, is applied, then the metal sample will stretch, and that results in a strain, which is a change in the length of the material divided by its overall length. At first, when you start stretching the sample, the deformation is elastic. So if you were to remove the stress, the crystal would snap back to its original length. But once you get past the yield point, the dislocations in the crystal begin to slip, and this plastic deformation results in a permanent change in the length of the sample. Notice the area under this graph is a measure of the energy that's stored per unit volume in the material. It's also called the toughness. Engineers always make that interesting. It's a measure of the strength of the material. So during plastic deformation, the dislocations will move until they reach the edge of the crystal. And you can see in this slide that the dislocation starts over here and it's traveling to the right until it gets to the edge of the crystal. The edge of the crystal is also known as the grain boundary in a larger piece of metal. So it's like there are several, well, thousands of crystals that all kind of intersect with one another at odd angles. And so the dislocation will move until it gets to the edge of the grain boundary and it can't go any further than that. Grain size strongly affects the mechanical properties of the metal. Larger grains allow the dislocations to move farther before they get to the edge. During the plastic deformation, the metal is more ductile, you can stretch it more, but it's not very strong. Smaller grains, however, make the metal stronger, but because the dislocations can't slip as far before they get to the edge of the grain boundary, those metals are less ductile. Now, during manufacturing processes, typically what happens is you need to take a piece of metal and you need to work it. You need to form it into a sheet or a wire, and during that process, the cold working decreases the grain size, and that eventually causes the metal part to become brittle, and if you continue to work it, it could crack. So to restore the ductility of the metal at periodic times during the manufacturing process, you need to anneal the metal. This restores the ductility of the metal by raising the temperature above its recrystallization temperature and then cooling it slowly to allow the grains to grow large. And you can see on the right side here, the large grains after annealing. Cold working increases the overall energy stored in the material, but it decreases the grain size. The annealing process is both time and temperature dependent. So the higher the temperature, the faster the material will anneal. The longer you expose it to the higher temperature, the greater the annealing effect. Now, because annealing is necessary and expensive, finding methods to anneal at lower temperatures or for shorter periods of time would provide immense cost benefits. Our investigation studied whether the introduction of a magnetic field could produce those results. So benefits of magnetic annealing, if possible. One is that it would decrease manufacturing costs due to the less expense for energy and less fatigue in the metal parts as you work. The other possible long-term benefit is that it might be possible to repair parts in place in the machinery by subjecting them to not as high a temperature, but using the magnetic field to essentially repair them in situ to save time and also be able to extend the lifetime of the machine.
Okay, so now I'm going to talk about what we actually did in the lab. And as Joe said, this is in industry. They want us to be able to do this. So we got all of our samples from actual industrial parts. This is a die used to make a part of a car, and it was very worked. And you can see how they have a whole label. There's a tiny hole there. But they cut our samples out from both sides where it was the flattest, and then cut us strips. And this was from our nickel sample. We also used aluminum and stainless steel, but they went from the sample. This is the only one we saw. So our samples came from actual industrially used parts, not just bought and used. So they did have some work stress due to them. So the first thing that we did is when we got our specimens is we measured and labeled and recorded. So we did this with our aluminum, stainless steel, and nickel samples. So
So we numbered each sample, recorded it, and measured length, thickness, and width of each sample multiple times. For the width, we measured the top, the middle, and the bottom for each one and recorded. And for length, we measured the entire length. And we made sure to record which one went for top, middle, bottom with every unnumbered specimen. We then stressed our specimen using an electromechanical tensile testing machine. It was the Instron 4507. And we stressed our samples to either 0.1, 0.3, or 0.5%. In order to measure the stress done, we attached an extensometer to the metal. There's a close-up view of it. We attached it to the metal so it could very precisely measure the small difference in pull when we pulled the metal, the distance that it stretched. We attached the extensometer to the specimen using rubber bands, and when you cut them off, they flew everywhere. We had to measure the length between the two grips, record loads, and look at the extension after it was done. While the process was running, we got to see a live graph of what was happening to make sure there was no slippage of the extensometer. We also got to compare all the different trials to each other as it was going on in real time. We also had to record the temperature and humidity at each time because sometimes if you have an extreme temperature and extreme humidity, the metal might act differently than you would expect. Afterwards, we then measured again the length, the width, and the thickness to determine if there was a change. You would expect if you're pulling it, and especially if it undergoes plastic deformation, that it would lengthen, and it did. So it was neat to see immediately that something actually happened. And again, we recorded our results. This is where we ended with our aluminum and stainless steel samples, but we kept testing with our nickel. With our nickel samples, one of the first thing we did was heating. We heated and annealed our samples in a quartz clamshell furnace. We heated both at 400 and 700 degrees Celsius for varying amount of times to see what would happen. With a couple of our samples, we also heated while under a magnetic field as well. So we did 400 degrees Celsius and 9 Tesla for both 2 hours and 4 hours. And we used a 9-inch vertical bore 9 Tesla magnet from American Magnets. Inside the magnet, our sample was in this susceptor, which had a graphite inside and insulation around the outside. The reason we used the susceptor as opposed to just putting the specimen inside the magnet is we were using induction heating, and if we did not put them in the susceptor, then the sample would not be heated evenly. Again, after we got our samples back from annealing, we re-measured. We re-stressed to the same percentage that we did previously, and then we measured again. And then Josh.
I spent the better part of 2 weeks trying to learn the vocabulary, so this picture is very appropriate. However, like Rhonda, we come to the part where we're going to look at some of the graphs. And so I'd like to think I am okay when I'm dealing with, okay, the x-axis represents and the y-axis represents. But when it comes to what's actually happening out in the real world, the material science part, that was kind of hard to figure out. So just to give you guys some perspective, like she mentioned, we did somewhere we just used heating only, okay, in the furnace. We're going to see some where we had magnet only, and we're going to see some where we have a combination of the two, heat and magnet. So what we're looking at in all these graphs, what you'll see is the blue curve is always going to represent our original stress versus strain, so that we can do some comparisons of the conditions that we put our specimens under. In this one, like I said, we went through heat only, and this one was 1 hour at 400 degrees Celsius. After restraining it, we found that it was, I'd say, somewhere between partially and fully annealed. So we have weakened this specimen. And so from this point forward, that should give us some sort of reference as to what we're looking at with each of the two, but maybe even three curves. Okay, so here we went with, there are two different things happening here versus the original. Now this goes back to the group back in June. We wanted to wrap up some of their samples. They had taken some just under the magnet at room temperature, and it was sort of discovered that there wasn't much effect here. However, we may have strengthened it just slightly, but when we came back and put it under the heat, we noticed that we had increased the yield versus the original, and I suspect again because it had already been stressed pretty significantly after its original test. So let's continue on and now, here's where it gets a little more interesting. The first thing, again, this is still with the June group. We only went 10 seconds in the same magnet. Again, we've strengthened it maybe just slightly. However, we came back and we were told to, let's go with a heat test at 700 C for an hour. And as Jeff mentioned, you know, at this low curve, we're seeing a very weak, as Edgar said, we're basically looking at a piece of chewing gum at this point. When we pulled it out, it was essentially white. So it didn't really take anything to strain it or stress it any further. And we just saw some other things. There's 4 hours at 700 C. Okay, basically the same effect with the green curve with just the magnet. Okay. All right. And so this was just our testing from this point forward. We decided we want to go with just 8 new samples where we were the only ones that had anything to do with it, just to look at some specific things, because now we wanted to put magnetic field and heat at the same time. So what we have seen, let's look at the top one, magnetic field and heat versus just heat there in the lower right. We see what ends up happening is we're getting a higher yield point, which is sort of opposite of what we were hoping for. You know, the process is that the magnet would help anneal. However, it kind of had the opposite effect, which essentially is something that we would note when we go to further testing. As you can see, in the lower right, we just had the heat. So the magnet did have an effect beyond just the heat, but it had the reverse effect of what we were hoping for. And the next one is just kind of shows, emphasizes that again, kind of the fine detail of each of those horizontal lines, and see that the upper left one still shows that, makes that same point. All right, the next set of variables we want to look at is heating at 4 hours under the magnet at 400 C versus heating at 2 hours at 400 C. And from these graphs, we saw that the time difference was not that significant. So whether it was in there for 4 hours or 2 hours, it didn't have as much of an impact. We did that test a couple of different times. We see the same result twice. So some of the things, you know, as far as the connections, we sat and discussed how we would take this back to our kids, back in our own schools. You know, just that whole idea of an experimental design, whether it's, you know, in a math situation, you do that with my 80 stats kids, of course, in science all the time. Being able to formulate a question, control your variables, that was the biggest thing that we kind of had to do to, you know, because there are a lot of variables, time, temperature, magnetism at different levels. So controlling those variables was very significant. And I assume, and I'm sure, as going forward here, that's what they'll look at as they work these different alloys when they deal with whether it's the nickel, the stainless steel, the aluminum. They'll start to fine-tune those variables that we were working on the past couple of weeks. Precise measurement, She showed that we were, measuring more than I care to do in a 2-week period, and writing more. I think his hand cramped up every day. Documenting everything, we would definitely learn that from Chris. Predicting outcomes, kind of what did we expect, and unfortunately, but fortunately, we kind of had the opposite outcome of what we thought. And then questioning those results. So again, where do we go from here? Collaboration. I mean, we had to, we were, this cramped up space right here, trying to do all of these things that we had pictures of. So working as a team, much longer. We may have tore each other's heads off, but we began to, we were working well together. We found a system. We went from, those exchanges of one specimen to another. We're starting out around the 20 to 30 minute range. I think we had that nailed down around 5 minutes these last couple of days. So we got more efficient. And we want to teach our kids to learn to be efficient, working in groups together. Obviously challenging ourselves outside our area of expertise. I think everybody sort of experienced that a little bit, except it sounded like Chris did exactly like her class was. And then knowing when to ask for help. I mean, I think we had to do that all the time. Sometimes we had to run across the street, make phone calls, send emails last minute, meeting deadlines, and working under pressure. Okay. So, yeah, I mean, that's why we're sitting in the back early. We're finishing up our project. It was fun, though. It was worth it. I mean, as a math person, I'm fascinated by physics especially, but also, chemistry. I've had an amazing experience doing this, just like all of us had. And so I'm looking forward to getting back in my classroom and I'm going to a new school, but I'm looking forward to meeting the chemistry teacher, the physics teacher, and seeing how we can work together. There's an engineering lab like you would teach. So I'm anxious to be a part of something like that. Do you want to talk about classroom connections? Or your own experiences?
So I'm trying to definitely, this past year we implemented a couple inquiry labs into our curriculum, but they were very boring because we were nervous about giving a little more free rein, I would say, to the students. But seeing this week, definitely realizing that it's okay to let them work together more, and they can, and definitely trying some of those labs that maybe I've never even done, and being okay with that, and letting them challenge themselves along with myself. So that's definitely something that I want to take. So thank you.
My classroom is very inquiry-oriented already, but I think the thing I take away from this week is just the way when you go into the lab at ORAU, everyone's willing to help you. I don't think it was just because we were teachers when they took pity on us. I think it was something that you saw, like people would walk in the door from another lab, and Chris was always up there ready to help them. What do you need? Okay, we can take care of that. We can do that for you. And that was great to see. And I really want to make sure my students understand that willingness to help other people, when you have the time to do it, is a great thing to do. And we definitely have some major thanks here. Edgar was kind enough to take something like 4 hours out of his each week with us to sit down and talk about the structure of materials, the properties, exactly what we're looking to do in this experiment. Really made it easy for us to know what we were doing and who to ask when we needed help. Chris, we spent probably 90% of our time in Chris's lab. And he, if you work at RNL and you don't know Chris, I don't know why, because he's been here 40 years and he has great stories. His father worked here during the war. And Bart Murphy was terrific. He helped us in the magnet lab. We got to take our metal samples and try to turn them inside the magnet. It was fun. And Jackie was actually in our lab group about half the time. She would drop in and see what we were doing, And she was kind of our bridge with the teachers that had come before because she knew what they'd been working on as well. So thanks to all of you.
And they were in the thin films and nanostructures group. And their mentor was Madam Lee, who is sitting right here.
Okay. I promise we will be brief. Okay. Like I said, we are in the thin films and nanostructures group. And I'd like to introduce my colleagues. First, I'm Phyllis Friello. I'm from Baltimore, Maryland. I teach at the Baltimore School of the Arts. We also have with us Haridas Chandran from Belfry High School in Belfry, Kentucky, and Judy Haggerty from River Edge High School in Woodstock, Georgia. We are all public high school teachers teaching physics, environmental science, computer technologies, and engineering. This is our mentor team. We are really fortunate in that we have not only one mentor, but a whole team. This team has been amazing. They have been extremely patient in answering questions, everything from why are you using argon and what exactly is that going to do to, can you explain to me how you actually pin down a vortex island? And they have been wonderful. And because we've had such a great team, we've been able to participate in quite a few projects from beginning to end, and we get just as excited, I think, as they do when something comes right, and then when something doesn't quite go as right, we're like, okay, well, what are you going to change? What is your next step, and why are you thinking that way? So it's been amazing. So basically, an introduction to the group, a primary focus of this group is to address the challenges in advanced energy technology. And to do this, they conduct the essential research to discover, understand, and manipulate matter on the atomic scale, thus the nanostructures. The group's research includes not only fundamental physics, as well as applications in nanoelectronics and energy technologies. And I was trying to think of a way to put this in words to really express what they're doing, and I didn't have any, so I just took somebody else's. Actually, a Dominican priest, Henry Dory, back in 1881, who wore Citius, Altius, and Fortius, which you might recognize as our Olympic motto. And I was thinking, okay, what we need, or what is the future of this, is faster processing speeds and capabilities. I had the good fortune to be at the U.S. Space and Rocket Center at Space Academy last year, and I was taking a look at the Saturn V rocket, and inlaid in one of the stages were the cables, the computer infrastructure used to service the Apollo missions, including Apollo 11 that went to the moon. And I was there taking a picture of it with my cell phone that had greater speeds and capabilities. And now, we use that to take pictures of our cats and send it somewhere. So that's the faster. The higher, I think a lot of us have probably seen the YouTube videos of the frog being levitated above a superconductor, and also I believe there's one with a magnet at Tel Aviv where it's being levitated with a track continuously. It's put into the shape of a loop. And while those are fascinating, the applications for those superconductors are not really practical for common use because of the incredibly low temperatures that are necessary to make them work. So the search is on for a high temperature, meaning hopefully room temperature, superconductors. And then the stronger part comes into play, where to complete these tasks, there is a great need for innovative structural design of materials with extensive, extended capabilities. Now, our group carries out this research by designing and developing new materials on the atomic scale, actually manipulating the tiniest aspects of these materials and then evaluating them and characterizing these materials. They understand the physical mechanisms in play and thus laying the foundation for the development of high-efficiency energy systems. Now, like I said, we were very fortunate to be involved in several projects, and a lot of these involved the following: material development and characterization. The first being photolithography, the process that's actually used in the fabrication of these films and the materials. Then they had to evaluate and characterize the surface, and to do this they used a couple of different methods: the atomic force microscopy, and then the bulk crystal characterization via X-ray diffraction. Our specific project that we're going to talk to you about tonight is we were tasked with putting a film on a silicon wafer, and of course we got to select a logo to print upon that wafer. And then what we did is we followed the characterization methods of looking at them in two different ways: the atomic force microscopy and the bulk crystal characterization by X-ray diffraction. And Haridas is going to give you a taste of that.
As she said, that we used 3 different techniques, and these are the final products from the research lab that we had, and this is not substantive any means. Okay, we just had some techniques that we thought we could learn and take back to the schools and learn something more about those. So the first thing what we did was to achieve this final product. We had to go through 3 different processes. The first one is called the photolithography. 2 different techniques that they can use. One is called the etching, and the other one is lift off. So for this, we used this particular lab, and you have it's called sputtering technique, and it's all used for gas fields. So there are 3 different gas that films that we can develop. One is titanium, gold, and your platinum underneath. So that's what the technique tells you something about how it's done. So we took a substrate, silicon wafer, and we used a process called, we used the sputtering, and the film was titanium and gold. The power that we used was 20 watts for titanium, and for 20 seconds to sputter. So we thought that the thickness of the titanium should be between 3.5 to 4 nanometers. We'll come across and let you know exactly how much it was. And the gold, we did the same thing with 20 watts, and we exposed it for 2 minutes, and we got 15 nanometer thickness. And for platinum, we used 40 to 50 microns, and for 8 minutes. And these are the original. These are originals that we can take home. So what we did was we used what is called, the second clause, we used what is called a photoresist. It's called 18, so 1818. And we put something on the film, on the top. And we masked the emblem using ultraviolet. And we developed that particular with CD26. So that removes the photoresistor, and we etched the gold by using Ka solution. So the product that we got was like this. And the next technique that we used we had one more so the next one is what's called the lift-off process for platinum that she did. She wanted to have platinum instead of gold, I don't know why. So she wants to be more precious. So that one, what we did was we had a silicon wafer, and we developed the envelope, and then we used a sputtering technique and put the platinum on it. And we developed that with using acetone, and we got the platinum. So, then what we want to know is, after making all these things, what are the information that we can get out of it? So the important one that we want to know is what is the thickness of the wafer that we put. So we use what is called the X-ray refractometer, and the principle is nothing but we follow the Bragg's law to these sine theta of the lambda, where you have the rays, the X-rays coming in, get reflected, and goes to the next lattice. There is path difference, and the path difference gives you a constructive interference. So from that they find out the peaks, so that they know exactly what type of material that you can get. But they can use the same thing, same refractometer, in a different way to find the reflectivity of the material, so that they can find out exactly what is the thickness of the material. So we used this one, and we found that the blue one is the experimental value, and the red one is the calculated value. And you can see that at one particular angle of incidence, that you see you have for a small angle you see a complete reflection, and because of the thickness of the films, you'll see what is called a fringe pattern that you can see when the angle gets increased. So this fringe pattern, if you can use a program and you can use a model, then we can exactly determine what is the thickness of the material. So we found that the gold is 41.06 nanometers thickness and titanium is 2.634. Remember the first one that I said that for that particular 20 watts in 2 minutes, we have a 15 nanometer thickness of the material. So we almost got that particular value, and we can say that this is a very good technique of understanding how much gold, what is the thickness of the gold. And also we can use what is called the atomic force microscope. So let me go back and then give you some the principle of AFM.
An AFM is an atomic force microscope. It lets you. An AFM is brought even closer to the surface. Increasingly repulsive forces cause the cantilever to bend away from the surface. Range attractive forces between the nanoscale. The close-range attractive forces between the surface and the tip cause the cantilever to bend towards the surface. However, as the cantilever is brought even closer to the surface, increasingly repulsive forces cause the cantilever to bend away from the surface. An AFM has a Z scanner that moves the cantilever up and down, an XY scanner that moves the sample back and forth underneath the cantilever, and a position detector that records the bending of the cantilever. The position sensor works by tracking a laser beam that is reflected off the flat top of the cantilever. Any bending in the cantilever will cause changes in the direction of the reflected beam. The position detector then tracks and records those beam changes. Using the detection method explained earlier, an AFM images the topography of a sample surface by scanning the cantilever over a region of interest. The raised and lowered features on the sample surface influence the bending of the cantilever measured by the position detector. AFM generates an accurate topographic map of the surface by using a feedback loop to control the height of the tip above the surface.
So we used this particular technique, and then we took one of these small portions here, and then we ran our AFM and found that this is the topography of that particular gold fillet that we have. And this one, you see the edges, you see it's gone because of the etching. You see underneath this substrate. And once you go from left to right, we see that the z-axis and the x at 10 micrometer, what you see is a big jump. That means that from the valley that goes to the top of the gold, that tells me exactly the length is 15 nanometers high. So with this principle, what we can say is, by using this particular ASM, we can easily measure the thickness of the film that we have. So these are the things that we study and we learn a lot of techniques. And I'm going to ask Judy to give us a summary of this particular film.
I have to say that, much to my group's dismay at certain times, I'm not a science person, I'm not a math person. I have a degree in English, but I've always had a fascination in solving problems. So 2 years ago, I asked to start an engineering program and got hooked into material science and engineering. So this came as a complete yay moment. And watching them in the lab, what I saw was them trying to make superconductors that we could actually use, which we can't right now. And they let us go through an entire process using their multi-million-dollar machines to let us see that it is actually possible that we can do these things. And it worked. The first time we tried this process, it didn't work. But Tony, God love him, our little PhD student, didn't give up. He got better and better at the process, and by the end, he was rocking it and taking care of it like he had done it all his life. We were able to get a personalized logo, and we know, you're all jealous. I have the brain on mine because obviously I'm pinky. So it helps us to remember what we got to see them do.
Okay, so our takeaways, I think one of the problems I'm going to have going back to school is seeing what I can kind of push aside in my physics curriculum to make room for a really long E&M unit. They don't need to learn relativity here. Just push that away. The logo that I chose was Saturn, you might have seen, and the reason I chose that was I had the good fortune a couple of weeks ago to go to another physics camp, not as amazing as this experience, but the most amazing thing at that physics camp is we set up the telescope one evening, and they said, we're going to look at Saturn. It's like, I've seen pictures of Saturn, I had one in my class, like, okay. But then I actually got to see Saturn, and to see just the incredible beauty of, and it's just this big, but the incredible beauty of that planet and its rings, and I was seeing it. It was fascinating. And I had a similar feeling in the labs these last 2 weeks of actually getting to see how these materials are laid down, and then with the different devices, with the different instruments, seeing the structure, seeing the surface, and how smooth really is when we're talking at that scale. I'm going to try and model some of the things with my supply closet that we did here, and work on a couple different labs, but I think the most important thing I'm taking back is that feeling of being fascinated by something, of being in awe of something, and having this incredible feeling of accomplishment when you finish an experiment, or when something goes wrong and you figure out why it's going wrong, and then do what you can to try and fix it, and then kind of continue. I have this great whiteboard in my room, and the kids come in and leave me notes all the time, or on their way out, they just do these magnificent doodles. This is one of the best things I've had being left on my board, is that science is cool. And that's what I bring back and reinforce to my kids.
What I'm going to do is, I teach physics for my seniors, and we do, for the last 9 weeks, we do a lot of projects, based studies, and Dr. Blackboard was kind enough to give us some materials. It is not superconductor, it is semiconductor, And what we're going to do is I'm going to ask my kids to study some material properties with different temperatures. And that temperatures can be used by looking at the chemical reactions too. So we can have some very high temperature as well as low temperatures. So we're going to try those things and see what my kids can take and also collaborate with the best scientists in this area. So I'll try my best, whatever I can do for my kids to better have a good future.
Okay, so I will probably not remember 95% of the chemical names of what I learned, but that's okay. Because what I've realized in the last 2 weeks is that scientists are people, and that they're really nice people who are always willing to help and explain, and like somebody else said, explain again and again, and finally use short enough words to where it actually penetrated my non-science brain, and I went, I get it! The photolithography is like photography. Okay, that makes sense to me. That's something I can show to my kids. I can also show them that you can be intelligent, outgoing, sensitive, and brilliant. And you can be a researcher whether you live in a bee house in Oak Ridge, Tennessee, or in Washington, D.C. It doesn't matter. What matters is that you want to know. You want to learn. And you're okay with going, it didn't work. Now what do I do? And for that, I thank Siemens, I thank Discovery, I thank Dr. Lee and all of the researchers. We all thank you in letting us have this opportunity. There would be no way to do this without you. And for the 19 people that I've lived with and worked with and complained at and everything else, thank you for putting up with me. My non-science brain appreciates everything that all of you have given me, and I promise I won't forget you. In the science, but I won't forget you. So thank you.
All right, our last group of the evening. All right, are coming to us from the Instrument and Source Division, and they're in the S&S Instrument Engineering Group, and Kevin Berry is their mentor.
And we'll only take about 45 minutes.
Yeah, so basically, Drew and everyone took all of our time. So we'll go really fast. We were planning to be quick anyway. My name is Emily Hill. I come from Los Angeles, California, where I teach science at John Marshall High School. These are my colleagues.
Hi, I'm Nate Wilson. I teach at the L&S STEM Academy, 30 minutes away, Knoxville, Tennessee.
I'm Mark Chabot. I teach in Thetford Academy in Thetford, Vermont.
Hey, go ahead.
All right. And we worked with our mentor, Kevin Berry, with the helium-3 gas detector group. So, first thing we did on our first day over at ORAU was we got to have a nice tour of the S&S building. And that is where a lot of research is being done using neutron scattering. And so why neutrons? What is special about neutrons that allows us to use them in that way? And first of all, neutrons have no charge, and so they're able to penetrate materials much better than, let's say, electrons would be able to. And they also have approximate wavelengths that are very similar to the spacing between atoms in different molecular solids. So they can also be used for imaging, as well as looking at the scattering patterns. Neutrons filled this gap that used to exist using different methods, and we see neutrons all in that yellow section. Whoops, that's why you can't use them. Thank you. So there's definitely this sweet spot that neutrons cover that was not covered necessarily before. And neutrons are used typically to give us better ideas of what atomic structures are. And we've been using neutrons since the 70s to get better ideas of what polymer structures happen to be. At the wonderful SNS instrument building, there are a lot of instruments that are all using the same neutron beam. That neutron beam just kind of comes out, and all the instruments are, arranged around it to make use of all of those neutrons. And what we found was that there are a lot of helium-3 gas detectors there. And our mentor, Kevin, is in charge of keeping all of those up and running properly. Without the detectors, the instruments are useless because we wouldn't get the information that's useful without those detectors. So even though Kevin may not admit it, he's a very important man. And at the Heifer facility, the high flux isotope reactor, we did have the pleasure of going on that tour with Kevin as well. And we found that almost all of the instruments there are also using helium-3 gas detectors. So it's extremely important that we understand how they work and what their limitations might possibly be. And so the question has come up with different researchers looking to use the different instruments that have helium-3 detectors with samples that are in a magnetic field. The question is, would that magnetic field affect the way the helium-3 detectors themselves are working? And that's an important question because the other type of detector is a lithium-6 scintillator detector. And we know that these particular detectors actually do see an effect when they're in a magnetic field, particularly because when the neutrons interact with your scintillating material and kicks out photons, those photons then are somehow changed into electrons, and those electrons then move down a photomultiplier tube where we get this cascade. And if the electron's path is being changed because they're introduced to a magnetic field, then not all of the input electrons, as they're hitting these different, let's see, can I make it work? Yeah, as they're hitting these different arrays and being multiplied, not all of those electrons are going to continue all the way through. So we're not going to have the output that's expected. So we know for sure that it would be bad to use this type of detector in a magnetic field. So I'm going to turn it over to Nick. There you go.
All right, I can't juggle multiple things at the same time, so I'm going to put this back in a second. All right, so this is what the helium-3 detectors actually look like. They come in all shapes and forms, but this was the actual set of tubes that we use for the experiment that Mark is going to be describing in a few minutes. And these are some helium-3 detector tubes that are actually installed in the Nomad detector tank at the SNS building. This looks kind of crazy. It looks like it's from an Apollo mission or something, but a neutron beam will be going into the screen and will strike a sample in the middle of the tank, and then scattered neutrons fly off in all directions and are picked up by our tubes. Now, how do these helium-3 tubes detect neutrons? How do they actually work? That's an important question because, as with the lithium-6 scintillator example, it will affect how the magnetic field interacts with the detector. Now, a neutron will fly into this tube, which is made of steel, and it's filled with helium-3 gas that's pressurized in our tubes up to 10 atmospheres. The neutron will strike helium-3 nucleus, and in a nuclear reaction will produce a tritium, H3, and a proton, H1. And then the reaction will release 764 keV of energy, which is distributed as kinetic energy among the reaction products. Now, these products will shoot off in opposite directions, and they will smack into other helium-3 atoms, releasing electrons and ionize those atoms. Now, these electrons will be accelerated toward a wire in the center of the tube, which is maintained at a high positive voltage. Now, these electrons will flow to one end of the tube or the other, depending on which one is closer. Well, it's resistive. It's resistive separation. Forget the term. But the charge will flow in both directions to preamps at the ends of the tube. And then the preamps will amplify and convert this charge into signals. And then these signals, the integrated area beneath the signals will tell us about the charge signal in both directions, and we compare the integrated areas in order to determine how far along the tube the neutron event actually occurred. So in this case, the neutron event occurred a certain distance farther to the left on that tube. Now, how will this change in a magnetic field? That is the question. Well, what happens to electrons in a magnetic field? They experience a magnetic force that is proportional to the velocity of the electrons, proportional to the field, and perpendicular to the direction of them both. Now, how is that going to affect the detection of neutrons? Well, these electrons that are ionized off the atoms, they will experience a force and they might move sideways through the tube, or they might move circularly through the tube due to combined effects of the electric and magnetic forces. And so what that means is that we might measure the neutron event to occur in the wrong position, or the resolution at which we can measure the position might change, or the height and shape of the pulse signals that we see at the preamps might also be affected. Now, this was one of the experiments that we did. We created a solenoid. Didn't create 8 Tesla like the group back there. 50 Gauss was as good as we could do with wire wrapped around a copper can. Thank you very much. 50 Gauss isn't a whole lot. I just had to look it up. It's about a refrigerator magnet. But if you're trying to study materials at an 8 Tesla field in a very small space in the center of the detector, it's going to fall off pretty drastically. About 50 Gauss at most, a meter away or however far away the detectors are. So we stuck a tube into the solenoid. This tube only has one preamp, so that it gives us a cleaner and more consistent pulse signal instead of being split into 2 different sets of signals. And what we found, I'm going to spare you all of the graphs and the data analysis and all of that. I would probably give a student an F if they did that for me, but for you guys, I think the time that we have, I'll just jump to the conclusion. Accounting for thermal effects due to the resistive heating of the solenoid, which also affects the detection characteristics, we observed no change in the pulse height distribution. And so Mark is going to talk next about another experiment that we did in which we investigated the effects of magnetic fields on the position measurements along the tube, as well as the resolution.
Okay, so here's where the rubber meets the road. First, so we're looking now at the back of the tubes. If we go back for just a second, this is the front. There are 8 helium detector tubes right here. And then we're going to be looking at the back of them right now in this next slide. And we use the high-tech suspension method of a rolled tape. And at the end of this, right there, we have our magnet, which is now behind the detectors. So we're going to get another look at this now. If we look straight down, we see there's a slit right here. Right? You see that little slit? Here's our magnet that's behind the slit. What we're looking at here are the detection tubes. This is number 1, and there are 8 of them stacked on top of each other right here. So over here we have a sample inside of the box of californium-252, which produces neutrons. Those neutrons fire into the detectors, and in a way that was described by Nick just a minute ago. And the question there, so then this magnet right there is applying a magnetic field. And so, of course, well, is there an effect by this magnetic field within the tubes on how the neutrons show up? We also changed the height position, of course, along these tubes using another high-tech method, the catalog. And the catalog has a certain unit called the page. And by adjusting the page, we can then adjust the position of the magnet along the y-axis. So much data was then collected. Now, we had to do baseline data without a magnet, for example, and then we had to put in many, many more trials. You can see these trials along here, and then some more trials after that. And we had to analyze them on a computer and look at the interesting outputs that come from all of that. And after all of that data was collected, including putting the magnet and using a Gauss meter and measuring the change in the field, making a field map of the magnetic field strength around the magnet at different distances. And what were our results? Of course, this is what we're all waiting for, because no one knew, right? Does a magnetic field affect the neutrons as they show up on these detectors? There does not appear to be any observable effects on neutron scattering detection using the helium-3 detectors we tested within the magnetic field strengths and orientations that we tested. Of course, there may be other kinds of helium-3 detectors and other kinds of magnets in other worlds that may have an effect, but within the parameters that we experimented with, there was no effect.
And
Which actually is a good thing that there were no effects. That means that people who want to do investigations using a magnetic field with their sample don't have to worry about the effect that it could potentially have on the detectors. We would like to say thank you to our fearless mentor, Kevin Berry, for being extremely gracious with his time and answering all of our questions. And showing us the value of using materials you have on hand when needed, even though you are in a multi-billion-dollar facility.
And he did not have a team of PhD students to dump us on, so he was stuck with us the whole time.
Yes, he took very good care of us. Gave us every tour that we asked for except for one, but that's okay. Did not get to see the tunnel. We'd like to say thank you to Siemens Foundation, ORAU, Discovery Education, and ORNL for letting us join you on this. We've had an amazing time, and particularly seeing how all the different departments have to work together, seeing how people need to be able to communicate between different groups. Kevin works with the gas detectors, but there are individual technicians that are each responsible for an individual instrument. And then there's all the research scientists that want to use these instruments. And so being able to communicate and work together is a very large part of what goes on at Oak Ridge National Labs. And that's definitely something that we're all planning to take back to our students, to remind them how important it is to have communication skills and to definitely build up those relationships so that you can pursue some of the higher level research that you'd like to do.
Yeah, so we did not prepare a takeaway slide exactly. So this is kind of off the cuff remarks, but I would just like to say that it's been a privilege to work with the teachers that were selected for this group. As a young teacher, it's hard to come by excellent role models. You know, I'm expected to be a role model myself for my students, but the teachers that I got to work with in this program, that I got to work side by side with, along with our mentor, you guys have been exceptional mentors for me and role models for what I can become as a growing professional in my field.
Yes, grown-ups. For me, I think just before we got here, we ended up with our school formed a partnership with a local Army Corps of Engineers Cold Regions Research Laboratory. And I was really wondering how I might work with them. I really didn't have any concept. And I think spending 2 weeks here with Kevin and my team, it really gave me a vision, I think, or sort of an inspiration about how we might form a partnership with that organization and really get the most out of it. So I'm really grateful for this opportunity to work with all of you. And also for the humanity, the human side of this as well as the science part. And all the fun that we had and the camaraderie that we had, including my intrepid roommate, Jay. Somehow Siemens managed to pair us up, and it was a wonderful experience for all of us. Thank you.
And really quickly, we wanted to acknowledge Julia Abbott,
This is her last group working with ORAU, and we really do appreciate all of your time and effort, and the fact that you went to great lengths to make sure that we were all well taken care of, well fed, and well entertained over the weekend. And we do sincerely wish you the best and hope that you enjoy your Boston adventure.
They're gonna cry. All right, before we start with the awards presentation, we have a little video that ORAU has made of the week.
The Siemens Teachers as Researchers, or STARS program, provides middle school and high school STEM teachers the opportunity to engage with top scientists and researchers on short-term research projects regarding current topics of national interest.
This is one of the best professional development opportunities that I've ever been a part of. It's been hands-on from day one. You're not sitting at a desk, you're not listening to speakers all day. You're in a group, you're working, and you're working with fellow educators of all different backgrounds and all different skills levels that all bring a different piece to the puzzle.
My favorite part has been the amount of interaction that we've had with the scientists here. I'm amazed at how gracious they were with their time with us, showing us how to use everything and involving us in their personal research.
This experience was life-altering. I mean, I am so re-energized. I can't wait to get back in the classroom and take the things I've learned. It's been a once-in-a-lifetime experience. I guess you could say it's something on my bucket list, to actually do real research, and now I've done it.
In addition to the research, ORAU provides additional educational enhancements throughout the program in the form of seminars, mini workshops, tours, and field trips to focus and expand the research experience.
The thing that's absolutely unique about Oak Ridge and this experience for me is that I feel like in some ways I'm part of the past, part of the history, as well as being part of the future.
You just take so much back, and the contacts you make and the people you interact with, so much more so than just the science. The science is awesome, but the people, you can't replace that.
I think what makes it unique is the fact that we get to work at a national lab. Other opportunities might be more local, but we're collaborating with teachers from around the country and working with scientists. And Oak Ridge has a really historical background that we get to be a part of.
This is a great 2 weeks for me. It's just a lot of fun to meet teachers from all across the country, and I really enjoy talking with all of the teachers just to kind of see how education looks different in their states.
I rarely take my students out to field trips just for various logistical challenges, but now that I've seen the value of actually being inside the lab and actually hearing about the science that the researchers do from them, now I really value getting out there, and I want to open up those opportunities to my students now too.
Part of what I'm looking at here is how people work together to work toward a common goal. And my students in the classroom really have a chance to develop their own understanding from the experiments, and it's very closely related to what we're doing here.
We have the ability to interact with top scientists in their fields. And these are scientists who are looking at the development. They're at the forefront of their field, and the things we learn about here are going to be in papers in a few months. So it's really exciting to be kind of at the forefront of these discoveries, these experiments, and these developments.
I think whether you are a science, math, engineering, technology teacher, you're getting to peer into other areas that it's going to help you understand how your subject connects to everything else, and how ways that you can prepare your students to be problem solvers and to not give up when something doesn't give you the results you expected, to be able to solve problems and
and try something else and go through that scientific method.
I'd say this was so far in my career the best professional development I've received in STEM, and I would certainly suggest many of my colleagues to apply. It's a great program.
If you get the opportunity, jump on it. It is definitely worth it. It's worth every minute. I mean, the days are long, but they're filled with so much valuable experiences that in my experiences, it's nothing. I've never done anything like this, and it's definitely worth it.
Well, how about that? Now you really are stars after all. Coming soon to a theater near you. Now, a special encore performance. All right, well, all the researchers in the room can now relax and take a deep breath. The hard work of the evening has been done, and it's time to celebrate. I want to start off by thanking you all for your presentations this evening. I hope you know how inspiring you are. Hearing your presentations and your stories about what the experience has meant to you makes me so proud. And I say that not just as a director of science education programs, but as the parent of an 8-year-old daughter who comes home from school every day beaming about how wonderful her teachers are. So I'm confident I can speak for other parents when I say how proud we are to have our children in your classrooms. So thank you for a job well done, and congratulations. You've heard us celebrate over the course of the evening the really remarkable partnership that makes this program possible and makes it successful. And rightly so, because it is a great partnership. But like all great partnerships, its success depends on the vision and the talent and the energy of a great leader. And our leader is Jennifer Harper-Taylor, president of the Siemens Foundation. And it's my pleasure to introduce her this evening. During the more than 10 years that Jennifer Harper-Taylor has been with the Siemens Foundation, she has had an impact on schools and teachers and students all across the nation. She has introduced tens of thousands of students and teachers over the years to genuine STEM experiences like this one. She's also been a tireless champion for diversity, both in STEM education and in the workplace over the years. And she's been instrumental in building the program portfolio at the Siemens Foundation. Today, she oversees an investment of over $7 million a year in innovative programs that are inspiring and supporting the next generation of scientists and engineers. Please join me in welcoming Jennifer Harper-Taylor.
Well, good evening, everyone. It's so nice to be here and have the opportunity to meet all the STEM fellows for this session of STARS, the 2014 STARS 2. We were here, of course, earlier this summer, and they were a phenomenal group as well. We never tire of hearing the experiences that you've had in the lab. At the Siemens Foundation, our mission is to engage and to activate active STEM participation by students and teachers through challenge-based learning. We have the exceptional opportunity to do that through a number of great organizations. Our partner, Discovery Education, they play a major role in the development of these programs, and of course, ORAU. We don't know what we'd do without you guys. When we talked about this program being developed 5 years ago, it really came from the essence of some great experiences that were already happening here, and the phenomenal relationship with Oak Ridge National Labs. Marie Westphal and her team have dived into this project wholeheartedly, found some phenomenal mentors, took the time to look at each of you as teachers and your individual contributions and what you could bring to the table to make this an enriching experience for all. And I say that from the perspective that we've got middle school teachers and high school teachers. We've got you from different disciplines. You're coming from different parts of the country. You're dealing with urban, suburban, and rural schools. So we wanted to bring a holistic approach to how we would look at how you can really take these experiences and translate them into teachable moments for students. And as educators, that's an important product that will be, given to students over and over again as you have them come through your classroom. And for that, you should be recognized for the time, the effort, and the expertise that you've gained here at this Experience the Stars. We really, really enjoy the fact that we're able to make this type of investment. It's one of those things where we have the rare privilege at the Siemens Foundation of investing in students and educators. And as I like to say, I get to do good things every day. And not everybody can say they get to do that. You guys impact lives every day. When you talk about the students that, are here at Oak Ridge doing, internships, and you weren't even aware of the fact that that was happening. Your students go on to do great things, and it's all based on the foundation you give them from the educational experiences that you have had and you're sharing now with your students. So again, for that, you should be recognized for your time and your effort, and it's our privilege at the Siemens Foundation to make sure that you have an experience like this, and we encourage you to go through many experiences. I heard some of you talk about going through different physics camps and going to space camps. Experience it all. If there are corporate and private and public organizations that are offering these experiences to educators, take advantage of it. I know that you had the experience to go through our Siemens medical facility to see some of the things that we're doing. There are facilities like that across the country throughout corporations. Take the time, reach out to those corporations in your community, and take your students in there. Let them see what a STEM experience looks like. Let them see that you don't necessarily have to have some 4-year PhD. Many of those employees working in that plant, they have 2-year degrees. We have 4-year degrees, we have researchers. There are all spectrums of how a student can have engagement in the STEM experience. So we encourage you to work toward making your students aware of that. Keep them excited and engaged, and get involved in as much as you can for as little as you can invest as far as money is concerned. If somebody else is paying for it, why not do it? And I think that we all know that an investment in you and an investment in students will ultimately impact society as a whole. And that is our hope, that we're able to make this investment into people who are energized, excited, and really, really wanting to make a difference in their community. And the teachers that you have come in contact would want to do that as well, given the opportunity to understand what you understand now. And that's that all of these programs exist for you to take advantage of. So I encourage you to keep doing what you do, to take this experience back and translate it into teachable moments in the classroom, and just always stay energized. The passion that you had in your presentations, I know they said you guys went long, but I thought you were great. I thought you kicked it off right, and you made everybody else come up to the charge, and all of you had phenomenal presentations. And I like the holistic view that you looked at everything from a people perspective, a team perspective, the engagement, the respect for the magnitude of the access that you've had. The fact that Oak Ridge National Labs gives this type of access to students and to educators is phenomenal. It speaks volumes to what they want to do to enhance the educational platform in the United States and to give access. Because obviously, these are investments that you as taxpayers make. And to know that so many things are out there that you can take advantage of and that you can use to really bring a more rich experience to your classroom, we want you to always be mindful that that's the case, because there's got to be a national lab near you. I encourage each of you to reach out to some facility, some university in your community, and try to make a connection so that you and your students can always have an enriched, free opportunity to learn more, do more, and engage more. So with that being said, congratulations on all of your great hard work, and it is now our privilege to recognize you. We think that each of you going through this experience are, of course, researchers now. If you weren't a researcher before, you definitely are now. So with that, we want to honor you and recognize you. by presenting you with a plaque that you can have in your classroom to share with your students and other educators. But more than that, we want to present you with a lab coat because you are officially now researchers. So I'd like to ask my colleagues to come to the stage. Julia, Dean, and Kate, will you come down so we can start our presentations? Thank you. So as we start, the first one coming up is always the person that gets to be the guinea pig. So with that, we're going to position you. You'll see where we'll stand for the photo. Our hope is that we'll be able to shake your hand. Kate's going to help you put on your lab coat. We're going to button at least the first button so we can have it nice and secure. And then Dean will have an opportunity to position you as well. And we're going to have you take a picture with your frame. And then our photographer is going to tell us how to lean it just so we don't have glare. Sounds good? Alrighty, so with that, we'll get started.
Derek Berger, Northern Garrett High School, Accident, Maryland.
We're going to ask you all to take off your name tags. You don't want to move the picture.
We didn't want to do it ahead of time. All right. Congratulations, Garrett. Ready?
Sir.
Yeah, thanks. All right. I'm going to bring you right over here. Oh, yeah, okay. You did it right. You're holding it right. Yeah.
So this is the first one, let me make sure we're good shape.
Thank you all very much.
Congratulations. Christine Brothers, Falmouth High School, Falmouth, Maryland, Massachusetts.
Okay, my sister teaches in Maryland. Thank you.
There you go.
You need an excellent stage. Just tell us before.
Everybody's gathered around good. Good, okay.
Congratulations.
Mark Chabot, Bedford Academy, Bedford, Vermont.
Come on, Dad. Congratulations. Sir.
All right, Derek, I'm slugging my gun. Congratulations, Mark. Congratulations.
Giridas Chandran, Belfry High School, Belfry, Kentucky.
Congratulations.
Phyllis Freelo, Baltimore School of the Arts, Baltimore, Maryland.
Congratulations.
Shannon Christina, St. Stephen's and St. Agnes School, Alexandria, Virginia.
Congratulations, Shannon.
Judy Haggerty, River Ridge High School, Woodstock, Virginia. Georgia.
You just moved me over.
Sorry.
Congratulations, Judy. Yeah, there we go. Thank you.
Megan Hart, Montgomery Blair High School, Silver Springs, Maryland. Josh Holt, Ottawa High School, Chattanooga, Tennessee. Say on my list, you're after
I was figuring it out, my name on it.
Emily Hill, South Marshall High School, Los Angeles, California.
Right there. Take care. Thank you. Thank you.
Looks good. Oops, got a reflection. Just hold it. There we go. That's good. Congratulations.
Catherine Hoover, Wiley High School, Wiley, Texas. High School, Middle School, Bunker Hill, Indiana. I'm sorry. Jay Kreisman, Hunterdon Central Regional High School, Flemington, New Jersey.
Wait a minute.
Viola Lavenhouse, Westfield High School, Chantilly, Virginia.
Westfield.
There we go. That's good.
Patricia McHugh, Isaac E. Young Middle School, New Rochelle, New York. Don Ramirez, Fort Stockton Middle School, Fort Stockton, Texas.
Thank you.
Rhonda Smith, Heritage High School, Maryville, Tennessee.
There we go.
Jeffrey Steiner, Arizona School for the Arts, Phoenix, Arizona.
There we go.
Kelly Weston, Seneca Valley Senior High School, Harmony, Pennsylvania. Last but not least, Nicholas Wilsey, L&N STEM Academy, Knoxville, Tennessee.
Here we go. Everybody's ready? All right.
Now, if everyone will come back to the stage for a group photo.
Half. So if you're shorter, bring it. But if not, if you're
All three levels
Because usually
Real close.
Okay, be careful. The ones that think they're tall, be careful, like this lady over here.
You got to do right here.
that thing tall. No, it doesn't matter. It has to
They have to back up.
Can't hide behind the signs.
Careful, somebody else beside the lady in the red, with the red hair. Stand.
Forward formation.
Somebody said,
Okay.
not you.
Jeff or me?
One of you guys.
I got a big gap here. Too tall. There we go. Everybody, I know we got a lot of photographers, but I'm the one that counts. So you look at me, okay?
Okay.
Well, I take one because I always have these. Okay, everybody look at it. Everybody's right here. She's posting to Twitter, so ignore her. Okay, everybody look right here. And don't, now look at Twitter over there.
like Kate, just saying. Those are so.
Let's see what I got.
It's the lab coats.
It's the STEM Academy logo, but
Hang on, I saw somebody close their eyes. I'm just kidding. Okay, now we've got it. You stay where you are. We're not going to make the mistake of letting you go because there's some other people that want their pictures made with you. So.
Facilitator.
Yeah.
Jim, come on. All right.
So.
And put your phones
I'm sorry. I'm tall, so I'm
And don't worry, everybody gets these pictures.
Wait, everybody is in like the world?
Everybody in the world. They don't, if you don't want anybody to know where you are, sorry.
Wait.
Let's see if I can see everybody. Looks like it. Everybody can say Siemens at the same time. I don't know if that's smiling or not when you say that. Let's see. Okay, hang on a minute. Take a different exposure. Try it again. Okay, here we go. Here we go again. Does anybody, Bo, do you want in there?
I'm getting mine right here.
Okay.
Is
that it?
No, the fun one.
No, don't. Yeah.
Everybody, I'm gonna do that.
The fun one, but it means it's not fun to get out of it.
That's right. No,
We're
fun people.
Yeah, we're not fun.
Creative science teachers. There's that pantograph
Are we ready? You're already figuring it out.
It's gonna be short.
Act natural.
We're doing our shoots.
Pretty good.
Are we good? No, we're not good.
Where's he going? Okay, that's good. Thank you.